So my lab works on Parkinson's disease and a range of things, from trying to understand what is actually going wrong so we can look for therapies to correct them, as well as other aspects in terms of how we could also help patients more directly with biomarkers, which I'll touch on at the end. So Parkinson's disease has a number of challenges, to say the least. This is just a graph that's going along with time, showing here at 0 years the time of diagnosis. It is a progressive disease as different parts of the brain start to degenerate or fail to function correctly, and that's associated as that time goes on with existing symptoms worsening and new symptoms unfortunately appearing. So there are some earlier symptoms prior to typical diagnosis by a neurologist. These are not common in all patients, but studies over time or longitudinal studies have found those aspects. And then by the time what's called a motor or movement symptom presents, that's typically when people go and see a neurologist or GP, and they may end up with their diagnosis. So what are some of the other challenges we have? Well, as you can see in this disease course, our 0 has a lot to the left of it. So relatively speaking, we're diagnosing it, clinically diagnosing it quite late in the disease course. So as I'll touch on at the end, we would like to be, as does the field, be able to, can we detect people much earlier on in the disease course? And if we have also got therapies that would stop or slow the progression, then that's the combination we're looking for: early detection, therapy to stop progression. And I say that because at this moment we have treatments for the symptoms of some Parkinson's syndrome symptoms, typically motor or movement with levodopa supplement, but where a lot of other symptoms that we have no treatments really for, or effective treatments. And lastly, we have nothing that stops this inevitable progression of the disease continuing to generate, even though you're on those symptomatic treatments. And why don't we have any of those therapies at this moment is because for a large number of patients, over 90%, we don't have at all a clear idea of what's causing this disease. And that's why, again, I'm touching on that. If we want to be able to help with people with the disease, we really have to find out what's gone wrong first. And so that's what my lab looks at quite a bit. So we use many tools in my lab, as we do here in the Garvan, but the one I'm talking today about is with more neurogenomics, which is looking at either the contribution of DNA or RNA and how that may be giving us clues as to what's going wrong, or actually maybe the base of what's going wrong. And so for this to happen, what we're trying to do is to say, okay, we have DNA, get my mouse going here, encoding all our information, and how is that information actually used by a neuron? Well, a sort of temporary photocopy is made, if you want to call it, called an RNA molecule, and it's a copy of the DNA, and then that information that's here in the RNA molecule, it finally ends up being produced into and acting as a template of telling the cell how to make a very specific protein. So if we have 20,000 genes, we could make 20,000 proteins. That's our Lego set as a human being, of basically using all those pieces that can come together in different combinations in different cells to give us, and they're obviously important in the brain. So one way of trying to understand what's going wrong in Parkinson's is to work out, in Parkinson's patients, are there some genes that we're making too much of, some genes we're not making enough of? Now the DNA stays constant, so the way we measure whether have we got too little or too much, rather than the Goldilocks, is to basically measure the RNA from each of those 20,000 genes. And we do that by a process called RNA sequencing. And this is, in essence, with the genomic technology we have at the Garvan, we can take RNA from people who have donated their brains post-mortem, both healthy individuals and Parkinson's patients, for which we're very grateful. And by doing the sequencing and counting, we can then go, for gene 1, there's more RNA of gene 1 in a patient. What does that do? Let's think about that. Of gene 87, there may be less. That's a very crude approximation, and the bioinformaticians in my lab would shoot me for that, but that's what I'm conveying to you. Where did we look of regions? I won't go into the detail, but obviously regions that are pertinent to Parkinson's disease. From these patients and healthy individuals' brains that had donated their samples, we looked at several different brain regions at different stages of degeneration. Now, before I talk to you about what did we actually find, what I want to convey to you is where we have that DNA going to RNA is, like in all things in biology, it's sort of a series of Russian dolls. You open one and go, I'm there, and find 5, surprisingly, looking at you. And this is the information of this was one gene is encoded on the DNA, and in color are the areas that will ultimately end up in a protein. And these are called exons, and they're basically interspersed by these spaces. And so what first happens is that you make a copy of this RNA that has an exact copy of the DNA, and then there's a splicing reaction, just like the old tapes where you could splice bits out. Except our splicing and our cells are smart to have secret symbols embedded in there to know where are these things called introns that should be removed, and where are the color portions that should be stitched or spliced back together. So this is called RNA splicing. And this is what is happening here, that splicing reaction will take out those introns, leave us with the different colored portions to encode protein X, and then the cell will make protein X. Now, in most cases, well, in many cases we would be teaching in our textbook that all the introns would be removed and all the exons would be left. But in fact, what we found is that's not quite the case. So from any one of your genes, you can make multiple different proteins by a process called alternative splicing. And this in color are the exons, the white are the introns, and so instead of basically just removing all the whites and ending up with this protein with those 4 different domains as they're called in it, you can have alternative splicing, where instead you may have, I will remove the red exon and only make that protein. You end up with 3 different proteins in this case from one gene. You may go, okay, who cares about that? But because whether they have the red domain or don't have the red domain will change, sometimes quite significantly, exactly what that protein does. Does it go to a different part of the cell? Does it bind a different substrate? Does it shake hands with a different protein? So you now have diversity there. Now, this is really important in neurons. So as cells differentiate and form neurons as you're developing, then this is quite important. And this is obviously a cartoon type version, but what you would find is that perhaps in neurons, you change that your alternative splicing pattern so the neurons make it so it does have the red exon. And these two are made in a non-neuronal cell. And therefore, you know that that red part of the protein is important for the neurons to be able to function. So what we did with all of our RNA splicing, all of our RNA sequencing to ask what's going on in Parkinson's brains was not just count is it up or down, but is the alternative splicing pattern changed? In the healthy controls, we would find they would have that extra domain that neurons need. But what we found, and this is not limited to one gene, but a number of genes, is that the patients didn't incorporate that red domain nearly as much as they should, and were making more of this protein. What's the problem with that? Remember, you need that red domain to be a neuron, to function as a neuron. So if you don't make it anymore, your neuron's probably going to have a problem maintaining its normal function. And as soon as you don't have normal function, you have a problem. So what did we find? Look, in today's time, I don't have time to go into that, but the genes and proteins that we found that were basically like this example here, not including their neuron-specific exons, they are all in areas of Parkinson's that are very interesting, that we were already interested in. Parts of how things move in the cell, other aspects of how they get rid of proteins they don't want, things that we already knew were sort of going on but didn't know why. And we need to not just identify them, but understand why so we know how to fix them. So this gave us a lot of spur to be very interested in this process by looking at the things that we found were being changed. But what really drove us was to say, okay, but why are you having these changes in alternative splicing? Because if we could understand that, we could adjust that problem at the top and fix all the underlying problems rather than work on one at a time down below. So, without getting into too many more details, is how does splicing work? None of us truly know. It is a very complex area, but basically part of this is that there are specialized proteins called RNA binding proteins, or RBPs, and their job is to identify, again, sort of signals that we've yet to fully understand that are flanking this exon we want to keep. And in essence, when that happens, that exon is kept in the RNA, and we end up with this, say, neural-specific aspect. So these RNA binding proteins recognize specific sequences that are present either side, and they go stay in, it stays in, you make the protein that the neuron needs. Now, this is where it ties in brilliantly with Robert's talk that was just preceding, is that some RNA binding proteins can form compartments, or in his really good analogy, they can form tents. And so this is, if you want to think about splicing, you need to be able to working right there in that little microenvironment. You want to form, do it quickly, move away, move on to the next thing. So we've got very quick up-down pop-up tents. None of the children tripping. So what this sets up is that that's both beautiful biology for us scientists who get into the puzzle and wonderment of, isn't that amazing how it does that? But also with complexity, there are problems if things go wrong. And this may well be that because these RNA binding proteins are capable of forming these tents or compartments, and maybe as we're aging, which is the biggest risk factor for Parkinson's and Alzheimer's, is that we're not taking the tents down fast enough, or something's there that's causing the tent to just never be able to be taken down. And it may be that, this is our diagram, and this is what we're investigating, is whether in this example where I've just named it RNA binding protein 1, it's might be forming an aggregate or a tent that can't be disassembled, so it stays in one other area and can't be shuffled over to another, and therefore it's not available for that splicing. Without that RBP1, because it's trapped in our tent structure, it doesn't preserve that special neuron exon, and instead the default splicing pattern takes place, making a protein that's not going to work well for neurons. On top of that, we also have, as Robert touched about, a protein called tau, but in Parkinson's there's a protein very prone to misfolding called alpha-synuclein. Some intriguing sort of over a sort of back of envelope conversations is whether, as this protein's aggregating, could it be triggering and interfering with that tent disassembly? So there are all sorts of very exciting options that we're looking at to try and find out, could the basis of all those changes from alternative splicing in patients, could they all arise because we're having proteins inappropriately not getting into tents or not coming out of tents in this dynamic compartmentalization that Robert talked well on. Just lastly, I wanted to touch more on the biomarkers, which are a way of finding, where are you in your disease state in other ways. You might be familiar with diabetes. You might have an HbA1c test where you go in, give a blood sample, they say you're a 5, great, you're managing your blood glucose, you're an 8, that looks kind of bad, we should see you. That's something they're measuring in your blood and giving you an indication of where you are in your disease. So that's a reporter function or a biomarker. So what was of interest for the RNA sequencing data we did in the brains was that we could also see that some of those changes are happening in the blood of patients. And this gives us the possibility that maybe we could find out about people's disease status in Parkinson's by looking in their blood and looking at some specific RNA molecules there to give us an indication of, do you really have Parkinson's, or are you progressing? And so that's a big part of what my lab looks at. So really just touch on this at the end. Here we are with this late diagnosis. We would much rather, obviously Robert with the tent decamper will give us a therapy earlier on, I'm hoping, but we need to be able to have a way of detecting to give it to people early. So that's what we're looking for, something much earlier in the disease course. And that's obviously before clear symptoms are occurring. So we can't look for clinical symptoms to give us that early diagnosis. They're not there. Instead, what we're looking at is, can we find RNA biomarkers? Well, we have some and we're testing them, that we could identify people at the time of diagnosis. And then through looking at other samples and some patient cohorts that go on to develop Parkinson's, can we spot those people much earlier? Because that would give us biomarkers to say, at 40, you rock up to the GP, have a blood test, and they go, all good, or hey, there's some early warning signs that given the blood RNA levels you have, you may be on the very start of Parkinson's. Let's talk further, especially if we have a therapy that may stop that progressing so you never develop symptoms. That's our hope and goal. So thank you very much for your time today. It's been a pleasure. And now we have the possibility for question and